My name is Chezi Missia, and this is the science behind COVID-19 testing. The coronavirus has changed our lives dramatically. It has flipped the world upside down in the way we live today. It can't be denied that the coronavirus has had a great impact on the year 2020. Nevertheless, the science behind COVID testing is very fascinating. In my presentation, I will be explaining what the coronavirus is, the tests that are available currently, and the science behind each one of those tests. The coronavirus is a respiratory disease that is caused by a specific virus called SARS-CoV-2. This virus causes a wide range of symptoms such as cough, shortness of breath, loss of taste or smell, and other flu-like symptoms. There are several different tests for the coronavirus. However, I will only be explaining a few of them in my report. The first test is a diagnostic test on a molecular level through a technique called polymerase chain reaction, or PCR. This can determine if one currently has the virus or not. The other test determines if one has antibodies that can be used to fight off the virus. This is called an antibody test, and this test can be performed in two different ways, a technique called ELISA, or the other one using a rapid test. Before I start explaining the process of how the PCR works, there is some terminology that one may be unfamiliar with, such as PCR, RNA, probes, and primers. PCR, or polymerase chain reaction, is a common technique that can be used to make millions of copies of DNA in a short amount of time, and can be used to determine if one has the virus or not. RNA is a single strand of transcribed DNA and carries genetic information for many of the viruses. Primers are just the short DNA sequences that starts the DNA replication process, and probes are basically primers but allow us to see the primer, usually with a fluorescent glow. Before the PCR test is performed, a nasal swab is taken to obtain the best viral RNA. Then this is placed into a tube with a buffer solution that extracts only the RNA, discarding any other junk. Along with the RNA, primers, probes, and an enzyme called DNA polymerase are also added to the tube for later use. Then this tube is placed into a machine called a thermal cycle. But before it goes through the whole process, the RNA has to be converted back into DNA or cDNA. This is because the PCR test is most stable using DNA sequences. SARS-CoV-2 virus is made of RNA, therefore it is less stable and more sensitive to the UV radiation. To fix this problem, scientists use an enzyme called reverse transcriptase, and this enzyme is able to copy all the RNA and translate it back into DNA, which then can be used for PCR. Now the process of PCR can actually begin. The first step is called denaturing. And in denaturing, this is simply separating the strands of RNA into two separate pieces of single-stranded DNA, using high temperatures to break the hydrogen bonds between them. The second step is called annealing or attaching, and the annealing process is done at a relatively lower temperature than the previous step because it allows the primers to attach by hydrogen bonding, and hydrogen bonding cannot happen at a higher temperature. But before this happens, probe DNA is introduced, and these probes are able to bind onto a small specific sequence present only on the viral DNA. Then the primers are introduced and will start at the beginning of the viral DNA strand, leading up to the last step called extension. The last step of the PCR process is called extension, and in order for this to work, the temperature is raised once again. An enzyme called DNA polymerase is introduced and will use that primer on the viral DNA strand to replicate the sequence, and as it is replicating, the DNA polymerase will hit the probes that are already on the strand and release a fluorescent molecule before continuing on to replicate. And this process will continue until millions of strands are replicated, and thus there will be many fluorescent molecules. If there is an active case containing viral DNA, there will be a large intensity of fluorescence given off by the molecules and will produce a glow. This will be considered a positive test, and if there is no glow, the test is considered negative. There are two different tests that can determine if one has the antibodies against the coronavirus. Antibodies come in two different types, IgM and IgG antibodies. IgM antibodies appear quickly after the infection, and IgG antibodies are in your blood for long-term protection against other viruses. The first type of antibody test is called a lateral flow test or a rapid test. In this test, a DNA sample taken from a nose swab is placed into a well that contains a buffer solution, and in that solution, it will slowly start to travel down the strip using capillary effect. Capillary effect is when the water will start to move because of the different forces. The first pad it will stop at is called the conjugate pad, and this is where some of the pieces of viral SARS-CoV-2 proteins and colored probes are located. If the patient contains COVID antibodies, the proteins will join together and continue to go down the strip. Once it detects the antibodies, it will either detect IgG or IgM antibodies. Based on the results, a single red line on the IgM will indicate antibodies, and the same goes for IgG antibodies. If antibodies were detected, a control line will appear, and this will determine how accurate the test was. The second antibody test uses a technique called ELISA, which stands for Enzyme-Linked Immunosorbent Assay. This test uses a small drop of blood, unlike the other ones, and the blood goes into a tube that is placed into a centrifuge that gets rid of all the red blood cells, leaving behind only a serum that contains the antibodies. Dishes that contain live SARS-CoV-2 proteins are also used for this test. The blood serum is added to these plates, and the antibodies will start to bind to the proteins of SARS-CoV-2. These plates are washed off to obtain a better view of which antibodies stuck. A fluorescent enzyme is also added to the plates to determine the different types of antibodies. If the test is positive, the antibodies will glow and bind to SARS-CoV-2 proteins. If negative, there will be no binding or glow. Using these tests and the ones currently being developed, eventually we could be on the other side of this pandemic.